Dear students, let us start with our second chapter, sexual reproduction in flowering plants. As in the previous chapter, I had told you the biological significance of the life is the reproduction. So, here in this chapter, we study about the reproduction in flowering plants. So, in flowering plants, the reproductive unit is a flower. So, first of all, let us study the typical structure of a bisexual flower. So, in bisexual flowers, the floral parts are divided into four whorls, and these four whorls are attached to the solan base called the thalamus. The outermost accessory whorl, which includes sepals, as shown in the diagrams, are called as the calyx, and the calyx is green in some plants or might be colored. Generally, it is green and includes the sepals. The second accessory whorl is of the petals, and these are generally colorful as shown in this flower. The bundle of petals consists the second whorl, accessory whorl, called as the corolla, and next to the corolla is the male reproductive whorl, which includes the stamens, and hence it is called as the androecium. So, androecium is the male reproductive whorl of a flower, and the next or the central most whorl is of the female reproductive organs called the carpels and is called as the gynoecium or pistil. In this diagram, all parts are shown very clearly: sepals, ovary, style, pistil, stigma, petals, anther, filament. So, all these parts are labeled over here. Study it properly and draw the well labeled diagram also. So, now coming to the functions of these four whorls, that is of calyx, corolla, androecium and gynoecium. First of all, calyx. As I already told you, calyx is the outermost whorl, which is green in color and accessory whorl. So, it protects the flower in a bud condition, and it is generally green, and hence if it is green, then it has a significant role in photosynthesis also. So, it is photosynthetic in nature. So, if it is colored, as I told, it might be colored in some flowers. So, if it is colored, then it attracts the insects for the pollination. These are the three functions of the calyx. First function is to protect the flower in bud condition. Second, if it is green, then it plays a role in photosynthesis also, and third, if it is colored, then it acts as a pollinator attractant. So, after calyx, next whorl is the corolla. Corolla, as I said, it is colored and also have the scent, hence it attracts the pollinators for the pollination, and sometimes the base of the corolla bears the secretory glands which secretes the nectar, and hence it is also meant for the attraction of the insects for pollination. So, it also provides the protection to the plant. So, after corolla, the next necessary whorl of the flower is androecium, which includes the male reproductive parts, and the function of androecium is the production of male gametes or the production of microspores. Microspores, that is pollen grains, which contains male gametes within the anther lobes. So, next whorl The innermost whorl is the gynoecium, which includes the female reproductive parts, and the male function of gynoecium is production of megaspores or eggs, fruits and seeds. So, these are the functions of these whorls. Now, coming to in this session, we only discuss about the male reproductive unit, that is androecium. So, male reproductive unit includes stamens, and stamen is male reproductive unit and consists of the following two parts: a filament, a long filament, and a bilobed terminal anther is present. Here in the diagram A, we had shown the filament and anther. So, coming to the structure of the anther, so in much of the cases it is two lobed, and the two lobes are separated with the tissue called connective tissue. Though the bilobed anther sac consists two theca separated by a septum. So, a typical angiospermic anther is bilobed and thicate. Every bilobes have the dithicates, two theca are there. In total, there are the four theca and two major lobes. The anther is tetragonal, means having the four sides, and each side consists four microsporogena, which includes the two lobes. So, here in this diagram we have the dehiscence of the pollen sacs, which is taking out the pollen grains. So, structure of microsporangium. A typical microsporangium appears circular and surrounded by four whorls. The four whorls of the microsporangium, namely as epidermis, endodermis, middle layer and tapetum. Here in the diagram B, showing the four layers. The outermost layer is the epidermis, the inner layer to epidermis is endothecium, and after endothecium middle layer, and after middle layer there are the inner layer, which is called as the tapetum. So, these are the four layer. So, coming to the anatomical aspects of epidermis. Epidermis is outermost single layered cells, which is protective in nature, and the second layer endodermis, it is again having the thick cells, and it is also a single celled layer, and it also helps in the dehiscence and protection of the microspore mother cells or pollen grains. So, third layer is the middle layer, and the middle layer is composed of 1 to 3 layers. As in earlier layers epidermis and endothecium, these are the single layers, but middle layer is having the 1 to 3 layers, and it also helps in dehiscence and protective in nature. The fourth innermost layer is the tapetum, and it has the cells of the tapetum has the dense cytoplasm and many nuclei, means the cells having the dense cytoplasm and multinucleate, and it provides the nourishment to the developing pollen grains. This is the function of the tapetum, which might be asked in the 1 mark questions. So, it is providing the nourishment to the pollen grains. The center of each microsporangia is filled with the sporogenous tissue or sporogenous cells, and at maturity these sporogenous cells leads to the formation of the microsporangium. So, next is the microsporogenesis. Microsporogenesis is the process of formation of microspore from the pollen mother cells, and the process leads to the formation of the pollen grains. The microsporogenesis includes the process of cell division, which is reductive in nature, and hence it is the meiosis. Each cell of sporogenous tissue in the microsporogenome act as a potential mother cells or microspore mother cell. Each potential mother cells undergoes meiotic division, means meiosis, leading to the formation of 4 cells and reducing the chromosome number into half, and these 4 cells are called as the microspore tetrad, and on maturity the anther dehydrate and the microspores separate from each other and leads to the formation of pollen grains. So, after in this diagram is showing the dehiscence of the pollen grains and releasing the pollen grains from the anther. So, coming to the anatomical aspects of the pollen grain. So, pollen grain develops from the pollen mother cells by reductional division, which is meiotic division. They are present, they represent the male gamete of a plant. Pollen grains are generally spherical in structure. They possesses 2 prominent wall layers. The outer layer is the axine and interlayer is the intine, as shown in this diagram. The 2 layers in the middle diagram B, 2 layers, the outer layer is the axine and inner layer intine, having the 2 cells, one is the vegetative cell and 2 male gametes are shown over here. So, the 2 walls of the pollen grain are axine and intine, and axine is made up of a material called as the sporopollenin, which is the most resistant organic material found in the nature, which withstands high temperature and high acidic conditions or alkali conditions. Because of presence of axine or the sporopollenin, pollen grains are preserved as fossils or may be preserved for many long time. So, the inner layer is the intine and which is composed of cellulose and pectin. The axine has a aperture where sporopollenin is absent, and this aperture where axine is absent is called as the germ pore. A newly differentiated pollen grains has central nucleus and dense cytoplasm. The protoplast mitotically divides into 2 unequal cells. The bigger cell is called as the vegetative cell and the smaller cell is called as the generative cell. Here in the diagram it is shown the 2 cells. One cell is the bigger in size, which is called as the vegetative cell, and 2 male gamete cells are shown. So, much of the angiospermic pollen grains are shed at this stage, means 2 celled stage. One cell is the vegetative cell and second cell is the generative cell. But in some cases this generative cell divides and leads to the formation of the 2 male gametes, and hence in some angiosperms the pollen grains are shed at a 3 celled stage. So, next it is all about the pollen grains. Last topic of today's session is the importance of the pollen grains. They contains the male gametes for the sexual reproduction. They are nutrient rich and thus are taken as food supplements. The pollen grains can be stored for years in liquid nitrogen at a temperature of minus 196 degrees Celsius, and this process of storage at very low temperature, that is minus 196 degrees Celsius, is called as the cryopreservation. They means pollen grains might cause allergies or bronchial infections and leading to the asthma and bronchitis in certain organisms like human beings and other animals. This is all over for the today's session. We have started